Dear students, today's lesson is about photosynthesis, which is the process by which green plants use sunlight, water, and carbon dioxide to make a glucose and release oxygen. It occurs in the chloroplast and provides energy for the plant and oxygen for other living things. And as I mentioned before, the photosynthesis happens inside the chloroplast. Let's learn about the main parts of this organelle. First of all, we have the outer membrane and the inner membrane. Both of them to protect and control what enters and leaves the chloroplast. Secondly, let's move to thylakoids. The flat disc, as you see, each one we call it thylakoids, where the light reaction happens. Okay, now the group of thylakoids, as you see here, we call them granum. The group of thylakoids, we call them granum. Now, the stacks, we call this a stack. The stacks of thylakoids, we call them grana. Okay, we call it grana, and that to increase the surface area for light absorption, so the efficiency of the work will be higher. Now, moving to last one, which is stroma. Stroma is the fluid around the grana, as you see, where the Calvin cycle happens, or the dark reaction, or the light independent reaction. Okay? And if you want to talk about the light absorbing colored molecules, which we call them pigments, they are found in the thylakoid membranes. These pigments absorb specific wavelengths of light energy, as it's clear on this graph. We have two types of a chlorophyll as a pigment. We have chlorophyll A, chlorophyll B, even we have carotenoids. Okay, and see on this graph, we plotted the wavelengths versus the percentage of light absorbed. Okay, let's focus with one of them. Let me take chlorophyll A. Okay, and you can see if we follow the white line, you'll find that the absorbance of the light mostly will be with the violet and the blue. Then again, they will reflect the green to yellow color. Again, the absorbance increase for the orange and red. Just try it with the chlorophyll B and you will see the absorbance. For example, if we want to talk about chlorophyll B, you will find the continuous line, this black color again, absorbance of violet, blue, and then reflecting part of the green, little bit yellow, then absorbance started again. Okay, so in general, chlorophylls absorb light energy most strongly in the violet blue region, as I mentioned, of the visible light spectrum, and reflect what light in the green region of the spectrum. And that explains why plant parts that contain the chlorophyll appear green to the human eye. So we should know that the pigment molecules are transparent without color. Only what you see is the reflected color only. And before starting with the details of this process, let me just summarize it for you how the photosynthesis has two stages. And let's look at this figure. Now, we have two reactions. The light dependent reactions, as you can see, this is the first part, which is happening in the thylakoid membrane. This is the thylakoid, this one disc, we call it thylakoid, as I mentioned, in its membrane. The goal is to convert the light energy to chemical energy. The light energy to chemical energy, as you see, an ADPH and ATP. This is the target from whom the light dependent reactions. Why we call it light dependent? Because its energy depends on what? On light. Moving to the second stage, which is light independent. That means these reactions taking its energy not from the sunlight directly. They will take it from whom? From the ATP and NADPH that's already produced by the light dependent. The second stage, we call it Calvin cycle or light independent. Calvin is the scientist who discovered this cycle. Happens where? In the stroma, the fluid region outside the grana. Okay, so again, it occurred in a stroma. Their goal is to use ATP and NADPH in order to do what? To fix carbon dioxide and produce glucose. So the first one, we use the water, and in the explanation of this stage, I'll explain it to you what is the target of using water that will end by the byproduct oxygen. And for the Calvin cycle or light independent, they fixed the carbon dioxide to glucose. Fixation here means changing the form of this chemical compound. Now let's start in details. The first stage, which is light dependent reactions or electron transport chain. As you can see, this is the stroma area. Here we are inside the thylakoid, we call thylakoid lumen. The first step, as you can see, is the light absorption in photosystem 2 and in photosystem 1 at the same time. Now, because of this light energy, what will happen with electrons? They will be energized. Excites electron in the chlorophyll that already in the photosystem 2 and photosystem 1. These high energy electrons travel through the electron transport chain from photosystem 2 to photosystem 1. As electrons move, energy is released, which pumps hydrogen ions into the thylakoid lumen, as you can see here, from the stroma to the thylakoid lumen, creating a proton gradient, the different concentration. Not only that, if you can notice here, the water molecules, what will happen with them? Water molecules are split, as you can see here, to produce oxygen as a byproduct, protons, hydrogen ions, and releasing electrons that will replace the energized electrons. This splitting, we call it photolysis. So again, splitting water molecules will release the electrons that will replace energized or excited electrons already, hydrogen ions or protons, and the oxygen as byproduct. And now the protonic gradients, the different concentrations of hydrogen ions, will it drives ATP synthase, this enzyme, to convert ADP to ATP. This process, we call it chemoosmosis. I will explain it in details, Now, after that, what will happen? We will look at the formation of NADPH, Electrons from photosystem 1 here will reduce the energy carrier molecule or electron carrier molecule NADP plus to NADPH. These two molecules, these two energy molecules, NADPH and ATP, we will use them in the Calvin cycle in stroma in carbon dioxide fixation. So, ATP is produced in conjunction with electron transport by the process of chemoosmosis. What is chemoosmosis? Again, the mechanism by which ATP is produced as a result of the flow of electron down concentration gradient, as you can see, from the high concentration to low. This is the thylakoid lumen and this is a stroma. The breakdown also of water is essential to this process. not only for providing the electrons that initiates the electron transport chain, but also for providing the protons, the hydrogen ion, which is necessary to derive ATP synthase during chemoosmosis. And now let's talk about the light independent reactions. After carrying from the light dependent the two energy carrier molecules ATP and an ADPH. In this cycle, we have three steps. The first one is carbon fixation, which will be happening by the help of Robesco enzyme fixing of carbon dioxide and changing to 3-PGA. The second step is the reduction. And the third step is regenerating again the 5-carbon molecule RUBP. In the first step of Calvin cycle or light independent reaction, 6 carbon dioxide molecules combine with 6 5-carbon compounds, which we call it RUBP, as you can see here. Here 5 carbon and here 1. Okay, to form 12 3-carbon molecules, we call it 3-PGA. Okay, now during that, the joining of carbon dioxide with other organic molecules, we call it carbon fixation. As you see here in the second step, what will happen? The chemical energy that is stored in the ATP and an ADPH that already we took it from the first chemical reaction, I mean the light dependent reaction, okay, is transferred to the 3-PGA molecule to form higher energy molecules called G3P, as you see here. So the energy here is in this molecule is higher than the energy of a 3-PGA. Now, ATP supplies the phosphate group for forming G3P molecules, while an ADPH supplies the hydrogen ions and electrons. Now, moving to the third step. In the third step, 2 G3P molecules, as you see here, leave the cycle to be used for the production of glucose and other compounds. So only 2 G3P. Now, in the final step here, I mean the fourth step of Calvin cycle, okay, the remaining G3P will regenerate the 5-carbon molecule RUBP that will join the carbon dioxide in the first step. Let me talk again about the Robesco enzyme, which its main role is at the very beginning of the cycle, where it fixes the carbon dioxide by converting RUBP to 3-PGA. By this way, we will finish the Calvin cycle. In another term, the light independent reactions in the stroma. And at the end, let me talk quickly about the impact of the carbon dioxide concentration and light intensity on the photosynthesis rate. As you can see here, more carbon dioxide, that means faster photosynthesis until the saturation level. But too little of carbon dioxide will slow the rate of photosynthesis. Now, moving to light intensity, more light, faster photosynthesis, again until the maximum capacity. But too much light can cause damage, which we call it photoinhibition. That's all. Thank you for watching.